Dear students, last time we have seen the processes of microsporogenesis as well as megasporogenesis in detail. Today we will have a look on these processes as well as we will try to understand the possibility of questions that may arise from this topic. So, for your understanding, I have make two separate points of microsporogenesis as well as megasporogenesis. First of all, we will observe microsporogenesis and then we will shift to megasporogenesis. So, this microsporogenesis process is the process where there will be a formation of microspore, or you may say the pollen grains from pollen mother cell or microspore mother cell. Both of the terminologies are synonyms of each other. You may call it as microspore or the pollen grain. It is same. So look at here, this one is the pollen mother cell which is diploid in nature. It means it has double set of chromosomes. So there will be two set of chromosomes. So this pollen mother cell is present in the pollen sac of anther. And you know that there are n number of pollen mother cells present in the pollen sac, and that's why it may form n number of pollen grains through the process of meiosis. So you will observe that this pollen mother cell will undergo the process of meiosis, that is the reduction division, through meiosis 1 continue with meiosis 2. When the meiosis 1 gets completed, it gives you a dyad, that is two-celled stage. Both of the cells are haploid in nature, and later on in meiosis 2 you will get 4 cells, and these all 4 cells are again haploid, making them as tetrad. तो पोलन मदर सेल से हमें 4 haploid cells मिलती है, and all you know this is the product of meiosis. So there might be a possibility that a crossing over may takes place in the chromosomes of these cells, and every individual pollen grain is different to each other. तो ऐसा हमेशा होगा कि जितने भी पोलन ग्रेन्स बनने वाले हैं, मानो 100 पोलन मदर सेल्स है, सो वो बनाएगी टोटल 400 पोलन्स, and all these 400 pollen grains are different from each other due to the possibility of independent assortment and crossing over. So once you got the tetrad, all of these 4 cells get separate and there will be a maturation takes place. This maturation is no more different from the process of differentiation, where each of the pollen grain, you may say immature pollen grain, develops to the mature one. It means that the pollen grain will have a cover of 2 important layers. One is intine and other one is exine. The outermost layer is called as the exine and inner layer is called as the intine. All of you know that the exine is made up of sporopollenin, one of the important chemical which will not allow pollen grain to damage from any alkali, acid, enzyme, etc. etc. So that's why pollen grain remain always protected due to the presence of the outer cover called as exine. And there will be an inner cover called as intine, which is just made up of cellulose and pectin. It is nothing but the cell wall material. So if you know that these pollen grains, they get differentiated into the functional pollens, and in this functional pollen there will be a mitosis that leads, and this mitosis leads to formation of 2 cells in one pollen grain. One of the cell is large and another cell is small. The larger cell is called as the vegetative cell and the smaller cell is called as the generative cell, and this generative cell is nothing but the gamete, that is male gamete. So if you observe, vegetative cells forms the pollen tube and generative cells forms the male gamete. Now there are 60% of angiosperms are available which have the two-celled stage of pollen grain, where one is vegetative cell and one is generative cell. Remaining 40% angiosperms are there where the pollen grain will have three-celled stage. One is vegetative cell and another two are generative cell. It means that 40% angiosperms, they have two male gametes in their pollen grain, and the 60% of angiosperms, they have only one male gamete in the pollen grain. So whatever it is, you must know that these pollen grains will travel, okay, reach to the stigma and performs the process of pollination. So that's why if you take one pollen mother cell, okay, like this, you will get 4 functional pollen grains out of one pollen mother cell, and that is the product. But in case of megasporogenesis, which is the next part of our discussion, we see again there will be a megaspore mother cell. This is the same like microspore mother cell, and this megaspore mother cell, that is meiocyte, undergoes a process of meiosis 1 and meiosis 2 and lead to the formation of dyad as well as the tetrad. So the process is same, whereas dyad will be the two-celled stage, both are haploid in nature, and tetrad will be the four-celled stage and all are haploid in nature. So all 4 cells that are formed from megaspore mother cell are haploid in nature. But out of these 4 cells, 3 will degenerate and only 1 become functional. Any one of the cell become functional. So that cell will become big, its cytoplasm will increase, its material present in the cell will increase, and that will be called as the functional megaspore. So from this story, you will clearly understood that one megaspore mother cell will give you one functional megaspore, whereas one microspore mother cell will give you 4 functional microspores. So we may say that the microspores are 4 and megaspore is only 1 in case of mother cells. So it is clear-cut indication that megaspores is only one that will be formed from megaspore mother cells. So if you observe this functional megaspore, it undergoes the process of mitosis and particularly the process of karyokinesis. So the haploid megaspore and the present nucleus in that megaspore will divide to form the karyokinesis. There will be total 3 subsequent mitosis will occur. As you will see in the first mitosis, the one nucleus divides to form the 2 nucleus. In subsequent second mitosis, you will see these 2 nucleus will divide to form total 4 nucleus, and further in mitosis third, that these 4 nucleus will divide to form the 8 nucleus. So altogether a functional megaspore contains total 8 haploid nucleus through karyokinesis process. It means a mitosis process. So there are total 3 subsequent mitosis that happens, which leads to formation of total 8 haploid nucleus. Now these 8 haploid nucleus get shifted. 2 of them will present in the center, which leads to formation of polar nuclei and the central cell. 3 will go to the chalazal end and they will form antipodals, and remaining 3 will go to the micropyle end and they will form the egg apparatus. Now, out of these egg apparatus, 2 were synergids and 1 will be the egg cell. So, if you observe the overall structure of this megaspore, you will see that there will be presence of total 8 nucleus and 7 cells, of which 6 cells are haploid and 1, that is central cell, is diploid in nature because it has 2 haploid nucleus, which makes it a diploid cell. तो ये तो हमें क्लियर हो गया. Now on this process there will be a possibility to have a question, and we will observe the question like question number 1. Please look at over here, question number 1. It says, to form 100 seeds, how many meiosis are required? Now if you want to have 100 seeds, then how many meiosis we need to have for PMC as well as for MMC? So you know that the answer will be 125. But if you got confused with this, please see below mentioned explanation for this question. As you know, as what we learned just before, one pollen mother cell will give you 4 pollen grains, and all these 4 pollen grains will be functional. So that's why for 100 pollen grains you need 25 pollen mother cells. So 25 pollen mother cells will divide by meiosis and will give us 100 pollen grains. Okay, now these all 100 pollen grains are viable and functional. If you go to the megaspore mother cell, then you will let to know that one megaspore mother cell will give you only one functional megaspore because the 3 get degenerates. So that's why you need 100 megaspore mother cells to form 100 megaspore. So 25 pollen mother cells will divide by meiosis. Total meiosis required 25 to form 100 pollen grains, and 100 megaspore mother cell will again divide by meiosis. Total meiosis required 100 to form 100 megaspore, and that's why in total 100 plus 25 we will require 125 meiosis to form the seeds. Now you will be in again dilemma that what seeds are doing over here, but you must know that one pollen grain fuses with one megaspore, I mean egg, to form one seed. Okay, so it is very easy that an ovule get converted into the seed, and ovule contain a megaspore and megaspore contain the egg. One megaspore contain only one egg, and one pollen grain that may contain one male gamete or 2 male gamete. But whatever the number of male gamete it is, only one male gamete fuses to egg to form the zygote, and zygote further leads to develop into the seeds. So that's why you need 100 pollen grains and 100 megaspores to form the 100 seeds. Okay, but to form the 100 pollen grains you need 25 pollen mother cells, and to form the 100 megaspores you need 100 megaspore mother cells, by which meiosis you will get the exact 125 meiosis to complete the 100 seeds. So that is how you can reach to this answer by solving the question like this. Okay, so there may be a possibility that they may ask you 100 seeds, or they may ask you 200 seeds or 500 seeds. Whatever it is, you have to go by this way to solve the answer for such questions. Okay, so question number 2 is, how many cells and nucleus are present in the functional megaspore? So just right now we saw that functional megaspore undergoes the process of mitotic divisions, that is karyokinesis division to give you total 8 nucleus. And all these 8 nucleus are haploid in nature, which further undergo the cell wall formation and cell membrane formation to form total 7 cells, of which 6 cells are haploid and 1 cell is diploid. So how many cells and nucleus are present in the functional megaspore? The answer will be total cells are 7 and total nucleus are 8. And you must know that these cells are divided like this, where antipodals are 3, those are haploid. Synergids are 2, they are also haploid. Egg cell is 1, it is also haploid. And central cell is 1, which is diploid because of the presence of 2 haploid nucleus. So in total you will get that 7 cells and 8 nucleus are present in the functional megaspores. So this is what we learned from the process of microsporogenesis as well as the megasporogenesis. Okay. So the next topic what we have to discuss, that how these pollen grains that are transferred to the functional megaspore and what are the ways of their transfer, that is the process of pollination. Okay. And before the process of pollination, we also have to observe the different types of ovules that are developed in different plants. Anatropous ovule is very much common ovule available in almost 90 to 95% of angiospermic plants, but there are certain different types of ovules that may be seen in case of angiospermic plants, and we will observe that also. Thank you very much for watching the video. I will share this PDF file with you. Thank you.